Not all traits determined by a single gene follow simple Mendelian inheritance patterns. Instead, several different patterns of inheritance have been found to exist. Some of these patterns occur when one allele isn't completely dominant over the other. In such cases, the classic dominant-recessive pattern doesn't apply. This brings us to degrees of dominance, where the relationship between alleles isn't as straightforward as Mendel once described. In Mendel's pea plant experiments, the F1 offspring always looked like one parent because one allele showed complete dominance over the other. In complete dominance, one allele completely masks the effect of the other. This explains why phenotypes of the heterozygote and dominant homozygote are identical. For some genes, neither allele is completely dominant, so the F1 offspring shows a mix of both parents' phenotype. Let's take a look at the cross between red snapdragons and white snapdragons. You might expect all the offsprings to be red, right? But here's the twist. They're not red or white, they're pink. This phenomenon is called incomplete dominance. The pink color in the flowers happens because heterozygotes make less red pigment than flowers with two red alleles. In incomplete dominance, the heterozygote has a phenotype that is intermediate between the phenotype of the two homozygotes, which is often known as the blended phenotype. Going back to our monohybrid cross, interbreeding F1 hybrids produces F2 offspring with a phenotypic ratio of 1 red to 2 pinks to 1 white. Also, as you can see, because neither allele is dominant, we don't use capital and lowercase letters to represent an allele. Instead, we use C with a superscript to denote the allele for flower color, CR for red, and CW for white. Now to recap, in complete dominance, one allele completely masks the effect of the other. In incomplete dominance, neither allele is fully dominant and we get a blend. But what if both alleles are expressed at the same time? Let's find out! Take a look at the human MN blood group. Individuals homozygous for the M allele have red blood cells with only M antigens, while individuals homozygous for the N allele have red blood cells with only N antigens. However, individuals heterozygous for the M and N alleles have both M and N antigens present on the red blood cells. This phenomenon is called codominance. In codominance, two dominant alleles affect the phenotype in separate distinguishable ways. Let's have another example to easily visualize and understand codominance. Take a look at a cross between a black chicken and a white chicken. In the F1 generation, all offsprings have black and white speckled feathers. The resulting phenotype is not the intermediate or the blend of the black and white phenotypes. Rather, both phenotypes are exhibited by the heterozygotes. Interbreeding F1 hybrids produces F2 offspring with a phenotypic ratio of 1 black to 2 black and white to 1 white. In this cross, we use C with a superscript to denote the allele for feather color, CB for black and CW for white. And that is how different degrees of dominance lead to unique inheritance patterns. Let's quickly recap. Incomplete dominance creates a blend of traits, like pink snapdragons. Codominance shows both traits equally, like black and white speckled feathers of a chicken. Both of these differ from complete dominance by showing the effect of both alleles, rather than one completely masking the other. The degrees of dominance is just one way to show how inheritance can get more interesting than simple dominant and recessive patterns. So, next time you think about genetics, remember, there's more to the story than Mendel's piece. Thanks for watching!